Here's a diagram of all the major anterior pituitary hormones. First, let's look at the direct-acting hormones. Growth hormone has a variety of functions, most notably controlling the amount of growth, so it has a positive effect on body size and growth in many tissues. Sometimes people have tumors in their pituitary gland, which leads to an over-release of growth hormone. These individuals are called pituitary giants. Prolactin is another direct-acting pituitary hormone. In mammals, it's important for the growth of mammary glands during pregnancy and regulating milk production. However, it has a variety of other functions too. For example, it's responsible for the brooding and parental care behavior in many kinds of animals. So the attendance at nests and with chicks, done by things like snakes and birds, is driven by the release of prolactin. Prolactin also turns out to be very important in land-water transitions in amphibians and in the saltwater to freshwater and freshwater to saltwater acclimatization that happens in salmonid fish when they migrate in and out from the sea. Prolactin is said to be responsible for the water drive. So in amphibians, it's high when they're in the water, and it decreases when they go on land. Similarly, in salmon, it decreases when they go out to sea because, again, I said this is sort of a hyperosmotic environment, more like living on land. And then it rises again when they come back to fresh water. The third direct-acting hormone is MSH, or the melanocyte stimulating hormone. Don't confuse this with melatonin, which is a pineal gland hormone, not a pituitary hormone. MSH is responsible for skin color darkening in animals like amphibians and reptiles and fish. There are four major tropic hormones. The first is TSH, or thyroid stimulating hormone. When TSH is released, it goes through the circulation to the thyroid gland in the neck, which causes the production of T3 and T4, two thyroid hormones. These have a positive effect on metabolism and growth. ACTH is the adrenocorticotropic hormone. This stimulates the adrenal cortex, the outer layer of the adrenal gland, to release corticosteroids. These then are important in the stress response and in regulating the balance between glucose and fat metabolism. There are two gonadotropins, luteinizing hormone and follicle stimulating hormone. These travel through the circulation to affect the gonads. Luteinizing hormone controls the production of testosterone and estradiol, the sex steroids. Follicle stimulating hormone, or FSH, controls sperm production in males and follicle development and secretion in females. Let's take a closer look at the pituitary's role in osmoregulation. This diagram shows the epithelial membrane of the collecting duct of a mammal. First of all, notice that there are tight junctions between each of the epithelial cells. This prevents water from leaking between the cells. A capillary is shown at the right outside of the collecting duct. Signals such as low blood volume or low blood pressure or high osmotic pressure indicates that you need to retain more water. Under these conditions, arginine vasopressin is released from the posterior pituitary. When it arrives at the epithelial cells of the collecting duct, it binds to a membrane receptor triggering a cyclic AMP second messenger cascade. CAMP activates a protein kinase, which acts on intracellular vesicles that store aquaporin II. This causes the movement of aquaporin II channels into the apical membrane facing the inside of the collecting duct. The aquaporin II channel now allows water to flow freely from the collecting duct into the cell. On the basal membrane of the epithelial cell are constitutive aquaporin III channels, which are always present. This then allows water to flow back across the epithelial cell out into the circulation, following the renal osmotic gradient that we talked about earlier. Once water balance is restored, the AVP signal is removed and the aquaporin II channels are resequestered into the cell. This is a diagram of a typical mammalian nephron. Notice on the right that the distal convoluted tubule comes back around and comes in very close contact with the renal corpuscle, which contains the glomerulus. Specialized cells on the DCT formed what's called the macula densa. Juxtaglomerular cells, or JG cells, line the afferent arteriole feeding blood into the glomerulus. These JG cells release an enzyme called renin. I'll talk about that in a moment. Under normal blood pressure and flow, there is low activity of the JG cells and they don't release much renin. In addition, the macula densa cells provide paracrine inhibition of the JG cells, further preventing the release of renin. The JG cells have the ability to measure the stretch of the afferent arterioles. This is a good measure of how high the blood pressure is feeding into the glomerulus. When there's not enough stretch, these release renin, and also under these conditions, the macula densa cells remove their paracrine inhibition of the JG cells. Renin is actually an enzyme, and in the blood it catalyzes the conversion of a peptide called angiotensinogen, which is produced by the liver, into angiotensin 1. Angiotensin 1 has 10 amino acids, but it's still an inactive form. A second enzyme called angiotensin converting enzyme, or ACE, is found on the endothelial cells of the circulation. These convert angiotensin 1 into angiotensin 2, which has 8 amino acids. Angiotensin 2 has a rapid effect and a slow effect. The rapid effect of angiotensin 2 is to increase vasoconstriction, which will increase peripheral resistance and increase blood pressure. It also leads to the release of antidiuretic hormone, or AVP, in mammals. It promotes thirst, and therefore it helps increase blood volume and blood pressure. The slow effect of angiotensin 2 is to increase the production of aldosterone in the adrenal cortex. Aldosterone acts on the distal convoluted tubule. Through nuclear receptors, it increases the production of sodium-potassium ATPase and sodium and potassium channels. Thus, aldosterone increases sodium and chloride uptake in the distal convoluted tubule, helping to retain water. What you can see from these examples is that the regulation and homeostasis of key physiological variables like blood pressure and blood volume and water and sodium happens through a complex of redundant steps. So the pituitary is involved, but also the kidney itself is involved. This is a table describing 4 major metabolic hormones. Animals don't typically feed all the time. They have intermittent feeding and periods of fasting. So they need to maintain fuel supply during these periods of shortage, and they also need to store nutrients just after they've eaten. The insulin-glucagon system is the short-term dynamic system that regulates nutrient mobilization and storage. These are both pancreatic hormones. Nutrients in the blood after feeding, for example, stimulate pancreatic beta cells to secrete insulin. Insulin promotes the uptake and storage of nutrients by a variety of cells. It's the only hormone that can lower blood glucose. The secretion of insulin is stimulated by high blood glucose, high blood amino acids, gastrointestinal hormones, and parasympathetic stimulation. All things that happen after feeding, and it acts in a hypoglycemic manner. In other words, it tends to bring down blood glucose. Glucagon is produced by alpha cells of the pancreas, and it's stimulated by low blood glucose, high blood amino acids, and sympathetic stimulation. Glucagon is an energy mobilizing hormone. It stimulates the breakdown of liver glycogen to release glucose into the blood. It also stimulates the mobilization of fatty acids from adipose tissue. So glucagon acts in a hyperglycemic mode, increasing blood glucose when it's low. So you can see why diabetes leads to a hyperglycemic state. If there's not enough insulin production, then glucose doesn't get taken up into cells and blood glucose gets very high. Epinephrine is produced by the adrenal medulla. It's released during stress, exercise, and fight or flight situations. It stimulates glucose release from the liver and the release of fatty acids from adipose tissue. Again, this is acting like glucagon in an energy mobilizing mode. Epinephrine also inhibits the release of insulin and stimulates the release of glucagon. Glucocorticoids are produced by the adrenal cortex. Typical ones are cortisol, cortisone, and corticosterone. These are involved in the long-term enhancement of glucagon and epinephrine effects. They stimulate the release of glucose and the production of new glucose. They also stimulate the release of fatty acids and the degradation of proteins. These are sometimes called stress hormones because they increase energy availability under periods of stress, but they have long-term negative effects, such as inhibiting the immune system. This figure shows what happens to blood glucose concentrations, insulin, and glucagon following a high carbohydrate meal. After the carbohydrate is ingested, you can see that blood glucose rises very quickly, and in response, insulin also rises very quickly. Glucagon shows the opposite response. One of the major effects of insulin is to stimulate the mobilization of GLUT4 glucose transporters from intracellular vesicles where they're stored to the cell membranes of cells to increase glucose absorption. GLUT4 is also mobilized by exercise in an insulin-independent manner. This is one of the reasons why exercise is good for type 1 diabetics that are insulin deficient, because in the absence of insulin to mobilize GLUT4 and uptake glucose, exercise can do it instead. The nervous system is also involved in the regulation of physiological systems. We're not going to cover in this course how neurons actually work, but suffice it to say that neurons conduct electrical and chemical signals at very fast speeds. So they offer an alternative way of regulating physiological systems from endocrine systems, which are more slow and long-acting. The nervous system, of course, also interacts directly with the endocrine system, either through the hypothalamus or by stimulating glands directly with nerves. Let's just consider how the vertebrate nervous system is organized. The central nervous system consists of the brain and spinal cord. These are central processing and control centers. The peripheral nervous system is composed of peripheral nerves and ganglia, which are groups of neurons together. The central nervous system receives afferent inputs from the somatic and what's called the autonomic sensory neurons. These convey information into the central nervous system. The central nervous system has two types of efferent outputs. One goes to the autonomic system, which consists of the sympathetic, the parasympathetic, and the enteric systems. Somatic motor neurons control your muscles. The somatic nervous system is your voluntary system, and it contains neurons that control your muscles. Somatic neurons originate in the spinal cord and go directly to muscles. They release the neurotransmitter acetylcholine, which stimulates muscle contraction. The autonomic nervous system is what we consider your automatic nervous system. It's not under voluntary control. The autonomic nervous system consists of two branches, the parasympathetic system and the sympathetic system, which act antagonistically to each other to achieve a high degree of regulation and feedback. Parasympathetic neurons originate in the central nervous system, and they synapse with other neurons in the ganglion near the target tissue. They release acetylcholine to the neurons in the ganglion, stimulating them to be active, and they also release acetylcholine at the target tissue. Neurons in the sympathetic nervous system originate in the central nervous system, but synapse on other neurons in ganglia that are very close to the spinal cord. These preganglionic neurons release acetylcholine, just like in the parasympathetic system. However, the postganglionic neurons release norepinephrine at their target tissue. In the case of the heart, the release of acetylcholine by the parasympathetic system slows down your heart rate. On the other hand, release of epinephrine by the sympathetic neuron increases heart rate. So you can see how these act in antagonism with each other. This is a schematic diagram of the autonomic nervous system in mammals. The sympathetic division is shown on the left, and the parasympathetic division is shown on the right. You can see that the preganglionic neurons of the sympathetic system originate in the thoracic and lumbar regions of the spinal cord. The ganglia are very close to the spinal cord. The sympathetic system has a role that stimulates what you would think would be important in a fight or flight situation. Therefore, it increases heart rate, constricts blood vessels, and dilates pupils. These are the kinds of responses you see when someone is in shock. It also relaxes your airways, so you can breathe better. It also stimulates the adrenal gland to release systemic epinephrine. So this explains why people who have asthma take Ventolin, which mimics the effects of epinephrine and opens up the airways, but also has a side effect of increasing your heart rate. At the same time, the sympathetic system inhibits the digestive system because this isn't important in a fight or flight situation. However, it stimulates the liver to release glucose. The parasympathetic system consists of a variety of cranial and sacral nerves. These generally have the opposite effects from the sympathetic system. They constrict your pupils, they stimulate salivation, they slow your heart rate, they stimulate digestion, and they constrict your airways. They also lead to the dilation of blood vessels, causing you to flush red rather than go pale. Study this diagram and know the effects of the two branches of the autonomic nervous system. This table summarizes the major effects of the parasympathetic and sympathetic branches of the autonomic nervous system. Generally, the parasympathetic system promotes restorative functions like digestion, relaxation, defecation, and salivation. The sympathetic system is the fight or flight system. It dilates vessels running to your muscles and constricts those running to your gut. It increases blood pressure and stimulates the adrenal glands to release epinephrine.